kailangan natin ng kahit anong organism para makasurvive. So, ang tatandaan nyo lang po dyan sa unang critical polymerase ay yung replication nga po is DNA from DNA or DNA into DNA. So, punta na po tayo sa pangalawang critical polymerase. Yung pangalawang critical polymerase po natin ay yung RNA-dependent DNA polymerase. Si RNA DNA polymerase po ay pwede din nating tawaging reverse transcriptase or RT. Also, ayun nga po, also known as RT. RNA-dependent DNA polymerase is a DNA polymerase enzyme that transcribes. What is transcribe? Yung transcribe po ay yung pagsisynthesize gamit yung existing nucleic acid or typically DNA as template siya, thus copying the genetic information katapos po. Balik tayo ulit. So, dahil alam na natin transcribe, balik po tayo. Si RT daw po, or also known as RNA-dependent DNA polymerase, is a DNA polymerase enzyme na nagtatranscribe po ng single-stranded RNA into DNA. This enzyme is also able to synthesize a double helix DNA once may condition po na yung RNA ay na-reverse transcribe. Reverse transcribe meaning reverse, kabaligtaran lang daw po nung transcribe kanina in a first step into single-stranded DNA. So yung tatandaan nyo lang po dyan sa pangalawa is yung RNA-dependent DNA polymerase or reverse transcriptase ay nakatranscribe ng DNA galing po sa RNA. And the last critical polymerase that we have is none other than RNA polymerase. DNA-dependent RNA polymerase are the RNA polymerase enzymes that transcribes RNA from DNA naman po. Meaning, nagtatranscribe po or yung nagsisynthesize gamit po yung existing nucleic acid or typically DNA as a template siya, thus copying the genetic information later. So, additional po ulit, sa RNA polymerase po, may tatlo po siyang klase. Si RNA polymerase 1, RNA polymerase 2, at si RNA polymerase 3. Punta muna po tayo kay RNA polymerase 1. Si RNA polymerase 1, yung nagtatranscribe po ng genes na nag-e-encode ng structural RNAs para po sa subunit of ribosome. Again, si RNA polymerase number 1, siya po yung taga-transcribe ng mga genes na siya namang nag-e-encode po ng mga structural RNAs para po sa subunits of ribosome. Then punta po tayo kay RNA polymerase number 2. Si RNA polymerase number 2 naman daw po, yung nagtatranscribe po ng genes na nag-e-encode naman po ng proteins as well as meaning kasama yung subunit of small RNAs. So, pag-compare po, si RNA polymerase 1, siya po yung taga-transcribe ng genes na nag-e-encode ng structural RNAs para sa subunit of ribosome. Tapos si RNA polymerase 2, siya po yung sa mga genes naman na nag-e-encode ng protein as well as subunit of RNA. So, last but not the least, si RNA polymerase 3 po, siya po yung taga-transcribe naman. Inakyaw niya na, madami to. Siya po yung taga-transcribe ng mga genes na nag-e-encode ng ribosomal 5S RNA, tRNAs, and again, a subset naman po ng another small RNAs. So, yun po, dito sa pangatlo, ang tatandaan nyo lang as a whole, si DNA-dependent RNA polymerase ay nagtatranscribe ng RNA from DNA at may tatlo po siyang klase na si RNA polymerase 1, RNA polymerase 2, RNA polymerase 3. Si 1 po, siya po yung taga-transcribe ng mga genes doon po sa structural RNAs. Sa pangalawa, siya po yung taga-transcribe ng mga genes naman sa proteins na may kasamang subunit po ng mga small RNAs. And si 3, siya naman yung taga-transcribe ng mga genes na nag-e-encode ng ribosomal 5S RNAs, tRNAs, and a subset of another small RNA. Yun lang po ang sakop ng polymerase. And pasensya na din po kung paulit-ulit kasi medyo po nakakalito kung tutuloy-tuloyin po dahil nga po marami pong undefined terms.
Salamat sa pagbibigay linaw, Mr. Alvin.
Salamat po sa
Kayo,
pakikinig.
kayo po, Miss Janine, may share po ba kayo?
Ang isi-share ko naman ngayon sa inyo ay ang tungkol sa prokaryotic host. Prokaryotic host cells. The bacterium Escherichia coli, or simply called E. coli, which looks like a gram-negative, rod-shaped, coliform bacterium that is commonly found in the lower intestine of warm-blooded organisms. And it fulfills its use widely in many cloning protocols. Take note of the word cloning. It means cloning is the process of generating a genetically identical copy of a cell or an organism. In biomedical research, cloning is broadly defined to mean the duplication of any kind of biological material for scientific study, such as a piece of DNA or an individual cell. E. coli has been studied in great detail, and many different strains were isolated by microbial genetics as they investigated the genetic mechanisms of this prokaryotic organism. In addition, E. coli, other bacteria may be used as hosts for gene cloning experiments, with examples including species of Bacillus, Pseudomonas, and Streptomyces. The process of transferring a DNA into prokaryotic host cell is called transformation. It means the genetic alteration of a cell resulting from the direct uptake and incorporation of exogenous genetic material from its surrounding called through cell membrane. Siyempre, kapag may prokaryotic host, meron ding eukaryotic host. Diba, Althea?
Tama ka dyan, Janine. Now, for our next topic is eukaryotic host. Palipat pa na slide. Cheska. What is eukaryotic host in the tools used in genetic engineering? Sabi sa PPT, eukaryotic host is preferred to produce human proteins since these hosts with complex structures or with distinct organelles are more suitable to synthesize complex proteins. Simplify ko siya. It is said here that eukaryotic hosts are more likely used when synthesizing complex proteins. Why? Because of its complex structure. Ano nga ba yung complex proteins? Complex proteins or conjugated proteins are two or more simple proteins in union but with more or other substances or organic compounds. Simple proteins, ito lamang yung proteins na amino acids lamang siya nilalaman. While yung conjugated proteins or complex proteins, ito yung proteins na may amino acids pero meron din siyang more substances or organic compounds. Now, sa examples natin, naalala niyo pa ba yung sinabi ni Cheska about proteins? If not, well, sabi niya, proteins are responsible for how an organism turns out. For example, our eyes. Sa akin, brown. Sa iyo, black. Sa kanya, brown din. Sa iba, blue. That is because the proteins inside our body, yung kulay niya naging blue, brown, black because of the proteins. Dito naman sa example natin in eukaryotic host, yeast. Yeast is a commonly used eukaryotic organism that is used in brewing and baking. And it is a non-pathogenic organism, meaning that this product cannot cause diseases nor it can harm other organisms. Sa yeast, di ba, in terms of baking, ginagamit siya sa bread, di ba? Nilalagyan ng yeast yung bread para mag-expand while in the oven. Kaya parang iniba niya na rin yung physical appearance niya. physical appearance ng bread nung nilagyan ng yeast. Another example is fungi, which is also a eukaryotic organism that has been used in gene cloning experiments. Yun lang po sa eukaryotic host. Thank you.
Tama! At kung may eukaryotic at prokaryotic host, syempre, may maganda ring host. At ako yon. Ang susunod ay ang selecting of small self-replicating DNA at plasmid map na ipapaliwanag naman ni Miss Jaisel.
Next slide pa po. Okay, so in this slide, we can see the illustration of plasmid. Of course, it is the plasmid map. But class, we don't need to tackle this anymore. It's because we just need to focus on the meaning of plasmid and the function of it. Okay, before we start to tackle it, I have a question. What is the most important part of bacterial cell? Anyone who wants to answer? Again, the question is, what is the most important part of bacterial cell? Okay, Mr. Saralta?
Ma'am, sa tingin ko po yung cell wall po.
Okay. Thank you for your answer. So, one of the most important part of bacterial cell is DNA. Doon sa bacterial DNA, nagko-compose siya ng two important part. It is the chromosomes and the plasmid. But, siyempre, yung topic natin is plasmid. Doon lang tayo magfo-focus. Ang plasmids are small, circular, self-replicating DNA molecules. Para mapalawak natin yung idea, explain natin. Yung plasmid kasi, since it's a small molecule, separate siya. Madali siyang manipulate, means pwede mo siyang makontrol, malipat sa another place, or pwede siyang matanggal sa isang cell to another. Pero, ang kagandahan niya is hindi namamatay yung cell life. Kasi remember, nasa bacterial cell to ha. Kasi ang sa iba, pag natanggal lang isang part, nadadamay na din yung iba. So, yung result nun, hindi na sila mag-function. In molecular biotechnology, plasmids may act as delivery vehicles or vectors. A vector is a DNA sequence that can transport foreign genetic material from one cell to another cell, where the genes can be further expressed and replicated. Plasmids are useful in cloning short segment of DNA. Ano ba ang vehicle? Di ba iyon ang ginagamit ng tao for transportation para makarating sila sa dapat nilang puntahan? Ganon din sa plasmids. Para mapadala yung genetic material from one cell to another or yung protein link from another target, they need to have the vector. Kasi di ba yung genes, yun yung nasa loob ng DNA. Ngayon, pag wala yung vector, hindi niya pwedeng matransfer yon. So, napakahalaga ng vector para yung mga genetic material or yung protein link is mapadala sa specific target nila. And also, plasmid can be used to replicate proteins such as the protein that codes for insulin in large amounts. Additionally, plasmids are being investigated as a way to transfer genes into human cells as part of gene therapy. Example, in a hereditary disorder where human cells lack specific protein, inserting a plasmid into the DNA would allow cells to express a protein that they are lacking. Kasi meron tayong hereditary disease na kulang sa specific protein. So ang papel naman ng plasmid doon, ine-insert niya sa DNA yung mga protein para maibalik niya sa cell na nagkukulang. At yung mga pwede niya i-transfer is yung protein link for hereditary conditions. 'Di ba yung ginagawa ng mga RNA protein, minsan yung mga disease na related to an absence of protein, gaya nung hereditary disease nga. Kaya yung sa plasmid, yung ginagamit na tagadala ng protein is yung sa nakulang sa cell, iko-combine niya yon sa RNA or DNA ng cell para masolusyonan yung problema. So para mapaiksi natin, mapadali yung definition ng protein link, siya yung nagdidikit doon sa mga kulang na genetic materials. Plasmids are also used in genetic engineering to amplify or produce many copies of certain genes. Yung plasmids daw, siya yung nagka-copy, nagpapadami, or yung nagpapadami ng mga certain genes. Thank you.
Nantok na ba kayo? Muna, please. Last na to, promise. Last na. Our last topic is methods to select transgenic organisms. Lipat mo na slide. Francesca. Isa pa. Yan. According to Farasa, not all cells will take up DNA during transformation. That being said, it is important to keep track of the cells that have transformed and the ones that have not. Sabi din dyan na most plasmids carry genes for antibiotic resistance. Antibiotic resistance are infections that humans and animals can get. Kaya sobrang important na ma-identify natin which cells have transformed and the ones that have not. Transgenic cells can be selected if it has the capability to make a new product. Transgenic cells, ito yung cells that are genetically modified, meaning na it has been through the process of genetic engineering. Kaya yung ibig sabihin naman nito na genetic cells can be selected if it has the capability to make a new product. If yung outcome ng transgenic cell ay new product, ayun na siya, genetically modified na siya. But if not, walang nangyari sa physical appearance niya, walang nangyari sa looban niya, walang ibang components, wala, yun lang siya. Ibig sabihin, something went wrong with the genetic engineering. Now, to identify the cells that has transformed, here's other ways of detecting the presence of other reporter proteins, such as the X-GAL and Laxis system or green fluorescence protein. The X-GAL and Laxis system, these are components that is very kailangan siya sa technique na blue-white screening. Blue-white screening and green fluorescence protein is very similar since they both allow selection based on color and fluorescence. Magsaselect sila ng cells based on color and fluorescence. Yung green fluorescence kasi, nagsho-show siya ng bright green fluorescence when exposed to light in the blue to violet range. Yun siya, similar din dun sa blue-white protein. Itong reporter proteins naman, ito yung proteins na ginagamit to identify the cells that has transformed and the ones have not. Sabi sa Google search, reporter proteins are encoded by reporter genes that is used in identifying the cells that has transformed and the cells that did not. Yun lang po sa methods to select transgenic organisms. Thank you.
Cheska,
Yes po, sir.
tama ba na generalization na?
May valuing pa po, sir.
Oo, tapos valuing na. Okay. With that, siguro, Cheska, puputulin ko na kayo kasi magti-try na.
Sige po, sir.
Okay. Now, mamayang gabi, isi-send ko sa GC yung list ng members ng group number 2. Isi-send ko rin yung i-report nila. Parang 3 pages lang yun. 3 pages lang yung report. So, ang gagawin ko naman, instead na PowerPoint ang isi-send ko, yung mismong parang lecture o yung document ang isisend ko, hindi naka-PowerPoint. So kayo mismo ang gagawa ng PowerPoint based from the lecture, no? So hindi katulad ng group number 1 na sinendan ko ng PowerPoint kasi inedit na lang na yung PowerPoint. Ang gagawin ng group 2 from scratch. From scratch gagawa sila ng PowerPoint presentation. Now, isang malaking hamon ito sa group number 2 at group number 3. Kung napansin nyo, ang taas ng level ng performance ng ginawa ng group number 1. Pasabog kaagad. And I would like to commend all ng members ng group number 1. Napakahusay. Napakahusay ng group number 1. Wala. Actually, kahapon, sorry for the comparison, kahapon, ang dami kong nasulat na comments doon sa reporters from the other section. Pero dito, kung makikita niyo pinagsusulat ko, meron ako ditong notes. Puro nakalagay halimbawa. Saan ba ito? Wait lang ha. Siguro pakita ko, no? Nag-joke ako dito eh. Nasaan yung camera ko? Wait lang ha. Sorry. Hindi ko alam kung meron, nakikita ba yung screen ko? Hindi. Sorry, wait lang. Ayan. Ayun. Ayan po. Dito, magaling. Ayan, puro magaling. Nakalagay dito, Cheska, magaling. Bricks, ganda ng bukas, magaling. Puro magagaling. So, kung napansin nyo, no, for me, ang baba pa ng ano ko nito, ang baba pa ng tag dito, ng expectation ko, pero ang gagaling. So, sobrang gagaling. Ang gagaling ng group number 1. Sobra. Wala akong masabing kumbaga negative, talagang pinaghandaan. Ako mismo may natutunan, naintindihan ko siya. Naintindihan ko, alam mo yung tinuro sa paraang maiintindihan ng lahat. Na-reach na yung objective. Di ba ang goal natin, kailangan mapaintindi yung lesson sa classmates. At yun ang ginawa ng group number 1. Ang ko lang, no, siguro meron lang akong isa ditong naisulat sa grupo ni Cheska na kumbaga constructive criticism. Medyo matagal lang yung motivation. Yung sa letters, yung sa Mobile Legends. Medyo matagal lang siya. Pero the rest, ang gagaling. Comprehensive yung discussion. Hindi lang basta binasa. Pinaliwanag. Pinaintindi. And that is, or those are the things that I will be looking for the next reporter. Kung hindi nyo kayang higitan, pantayan. Kasi yung next report mas madali. Actually, pinakamahirap na report to, yung group number 1. Yung group number 2, medyo madali. At group number 3, yung pinakamadali. So, challenge for group number 2 and group number 3. Ang tawag dito, kung hindi kayang higitan, pantayan. Nakita nyo kung gaano kahuhusay ang ginawa ng mga classmates nyo. Ako, hindi ako na-bore. Hindi ako na-boring sa kanila. Ang galing. Wala akong maitapon. Sa kanila. Napakahuhusay. Yun lang siguro ang masasabi ko, napakagagaling. And congratulations, Cheska and group number 1. Sobrang galing. Sobra. Okay, now, another thing is, dahil dyan, mamayang gabi, ipopost ko yung activities, performance number 1, 2, and 3 sa Google Classroom. Next week ang ating deadline. Next week yung deadline. Dahil dyan, ang Volta, yung reporter sa kanila, ah, tatlong activities yun, di ba? Performances 1, 2, and 3. Isa lang ang exempted sila. Pero dahil napakagagaling ng members ng group number 1, lahat ng activities ay hindi nyo gagawin. Performance 1, performance 2, and performance 3, accepted kayong lahat doon. Yun ang inyong pinaka-reward. Sobrang galing ng pinakita ng group number 1. Tapos, kailangan yung attendance. Siguro, ganito, ang attendance ay 3 bagay. Makinig. Makinig na 3 bagay before ko kayong i-dismiss. Una ay picture. Picture ng notebook, ng notes. That's 1. Klaro yun, no? Picture ng notebook. Ikalawa, magsi-selfie kayo, so picture nyo, tapos, i-edit nyo yung picture. Ayun, mag-swag post daw kayo. I-edit nyo yung picture, yung selfie nyo, tapos ilagay nyo doon yung anong natutunan nyo today, anong pinakamatak sa inyo. Halimbawa, may selfie kayo, tapos lagyan nyo siya ng text sa kahit anong parte ng picture. Halimbawa, lagyan nyo restriction enzyme, molecular scissors, halimbawa, ganun. So, kailangan mag-selfie, tapos i-edit yung picture, ilagay yung anong natutunan nyo today. Kung madami kayong natutunan, edi punuin nyo ng text yung paligid ng picture. No? Bahala na kayo mag-edit. Pwede namang kung walang ibang applications, pwede sa story, sa My Day. Di ba nasisave naman yun? Yun. That's number 2. And number 3, siguro, bigyan nyo ako ng grade ng group number 1 out of 50. 50 being the highest. So, 3 ang isisend sa akin. Picture ng notebook, picture nyo, na naka-edit doon yung natutunan nyo today. Tapos number 3, yung inyong score sa kanila. No? That will be your attendance for today. Tapos mamayang gabi, isisend ko yung members ng group number 2, i-report ng group number 2, and ano pa ba isa? Nakalimutan ko na. Picture, tapos isang bilhin na lang pala, no? Isusulat yung sagot. Hindi itatype, isusulat. Hindi nyo i-edit, isusulat. Inuulit ko, isusulat. Tapos, sir, sa third po kailangan daw. Wait lang ha. Sabi ni Mark Strelka, sir, sa third po kailangan po mag-message ko sa inyo ng grade po. Yes, tama ka dyan, Mark Strelka. So, ulitin ko lang, no, isusulat yung sagot sa performances 1 to 3. Ngayon, ang sabi ko sa Volta, kasi maraming, yes, out of 50, marami kasing umaabuso na next week na ngayon deadline. Nalilate pa. O ganito, kapag merong isang na late ng pasa ngayon, halimbawa hindi niya na ipasa ng Tuesday, ng deadline, the following or the next time na magpapa-activity ako, on the day ipapasa. Hindi na next week. The day itself. Inuulit ko. Ngayon, next week pa ang deadline. Pero, kapag meron pa rin na late ng submission, the next time na magpa-activity ako, hanggang alas 5 na lang ang deadline, the day itself. No? So, please, wag nang malilate. Kasi ang tagal na ang tagal ng extension. Okay. Then, next week, please prepare for a quiz. Ayan. Iniisip ko nga kung magkaklase ako next week eh. Kasi sa Volta, ang gagawin ko, magkaklase ako. Magkaklase muna ako, uulitin ko yun sa Volta. Uulitin ko. Ako na mag-discuss sa Volta next week before mag-group 2. So, tingnan na lang natin kung anong mangyayari next week. Pero mag-prepare yung group number 2. Prepare for an exam. Prepare for the next reporter. Job well done, group number 1. Congratulations. And I would like to congratulate din lahat kayo kasi yung nakita kong pagkukulang ng kahapon ay nakita ko dito ngayon. Lalong-lalo na yung cooperation nung klase. Very good. With that, thank you, Edison. See you next week. Bye-bye.
Thank you po, sir. Bye po.
Bye po.
Sige po, sir. Bye-bye
Salamat
po.
po, sir.
Bye po. Salamat.
Thank you po,
Bye po,
sir.
sir. Thank you.